Hello, everybody. Welcome to the patient-centric sampling interest group webinar. We're just going to hold on for a few minutes while everyone comes into the room. But thank you for joining us today. Okay, so the numbers seem to have stabilized. So let's get started. My name's Neil Spooner. I'm the chair of the patient-centric sampling interest group. I'd like to welcome you to the second in our series of webinars. You'll be seeing adverts for more of them going out through the rest of the year and next year. Just to move on a little bit, we couldn't have these webinars without the support of our partners and sponsors. So I'd like to thank all of these sponsors who've contributed to bringing this event to you today. I'd just like to tell you all, we're recording this event today that will be permanently kept on the evidence base and also will be linked from the patient-centric sampling interest group website. So your colleagues and your friends will be able to access this in the future. So I'd like to start off by letting you know about the Q&A. So if you have any questions for our speaker, Michael, please put them in either, preferably the private chat, but if they happen to go in the other chat, that's fine. Michael will be speaking for 40 to 45 minutes, and that will leave us some time for some Q&As at the end, which I will read out. And if we don't get them all answered, we'll try and get them answered by email or via our website after the event. So please just type your questions in the private chat. With no further ado, I'd like to introduce Michael Snyder, professor at Stanford University School of Medicine, and he's going to be talking to us today about transforming healthcare using big data and remote monitoring. Thank you all, and over to you, Michael, and I'll stop sharing my screen and
yeah, we'll switch over. Great. Well, it's a pleasure to be here. And yeah, as you just heard, what we're trying to do is actually transform healthcare with big data and remote monitoring. And yeah, I'm happy to answer any and all questions that you might have. So we think the healthcare system's broken. It's broken in a lot of different ways. First of all, it's really sick care that we mostly practice these days as opposed to healthcare, meaning we tend to treat people when they're ill rather than keep them healthy. But if you think about it, even when we practice so-called healthcare, the process is quite antiquated. Next slide. Typically, what will happen is you'll get in a car or a transport system somehow to go to a physician, usually at a very inconvenient time. When you arrive at the doctor's office, it pretty much looks the same as it's done for the last 40 years, maybe changed it with a few new gizmos, but for the most part, it's pretty similar. When you're there, they'll stick a needle in you, which typically hurts, and they'll draw a very large amount of blood. From that blood and a few other things they do in the office, they actually make relatively few measurements on you. And then from those measurements, if you go over to item number 5, they'll treat you based on population averages. And we think every one of these steps can be improved, particularly with regards to the step 5. If we get at the next slide, this idea about treating people based on population averages. Well, you've probably been told since you're little, well, certainly if you're on the Fahrenheit system, that your oral temperature when you put a thermometer in your mouth is 90.6. But if you actually look at the data that's out there, this is one of a number of studies, it's more like 97.5, first of all. I myself am 97.3. But the more important point here is there's a spread. That 25th quartile is at the bottom of the box, that's 94.6. And the 75th quartile is at the top, 99.1. And what that means in today's world, if your normal healthy baseline is 94.6, you go to a physician's office and they measure you at 98.6, they'll tell you you're healthy, everything's fine, what are you doing here? Go home. But I guarantee if you're up 4 degrees over your healthy baseline, you're probably not healthy, something's off. And so that's a big theme of our work. Know what your healthy baseline is so you can detect differences. And we exercise that quite a bit. So your health is influenced by many things. Next slide. Your genome is certainly an important part of that in the upper part. But all these other things, including lifestyle and exposures, impact your health. So exercise, going around the circle, exercise, food you eat, stress. We all know intuitively these affect your health, as do environmental exposures and pathogens. And we're in a world now where we can measure a lot of these, some pretty well, like your genome sequence. Exercise, you can measure pretty well with activity watches and things. Food's clunky. But even if you can't measure these precisely, you can actually measure their effects by doing deep data profiles on people. Next slide. So a number of years ago, we set up something, we call a personal omics profile, where the idea is that, starting at the top, we'll sequence their genome once. And then from their blood components, mostly peripheral blood monocyte cells, we'll measure their epigenome, transcriptome, proteome. And then from plasma, we'll also measure the proteome, cytokines, metabolites, and lipids. This is on top of measuring the microbiome, gut, nasal. These days, we now do tongue and skin as well. And we also do deep clinical testing and questionnaire surveys that go with this. We have some advanced tests we run as well, stress echocardiograms done on many of the participants, and glucose control measurements like oral glucose tolerance tests. And finally, if you look over left, we also have smartwatches, started on that about 8 years ago when these mostly fitness trackers. So one aspect of all this is to take deep data dives on people. And the other aspect is to do it longitudinally. So looking at the bottom, we're profiling a group of 109 people. We sample them every 3 months while they're healthy. And then if an adverse event comes along, like a viral infection, we'll take 5 to 7 additional samplings. And so we've been running this study for over 10 years, getting quite rich. And so you might say, well, why are we doing all this? Well, we're trying to understand what it means to be healthy, not what it means to be sick. What does it mean to be healthy? How does it change over time? How does it compare between different people? What happens at the earliest time of illness? And perhaps most importantly, from the healthcare perspective, can we use advanced technologies like genome sequencing, deep molecular measurements with mass spectrometry? Can we use these to better manage people's health? And on the next slide just shows that just from the first 3.5 years of following people, we had 49 major health discoveries. Now, I should emphasize this is an older cohort, 53.4. But these health discoveries, they were all made spanning a wide range of areas. So starting from the right, oncology, cardiovascular disease, metabolic disease. No one area was found. And diving a little bit deeper, if you go to the oncology, you'll notice we caught some with early lymphoma, 2 people with precancers, MGUS, smoldering myeloma, 2 people with serious heart issues, that RBM20, that's picked up from genome sequencing, another picked up by the wearables, and so on and so forth. And what's important about all this, again, is that these were all found pre-symptomatically, meaning people weren't showing symptoms. By doing this deep data dive, we actually were better able to see what was going on with their health. And so the way we like to think about this, next slide, is that if you think about your health as a 1,000-piece jigsaw puzzle, we're trying to measure 500 or 600 pieces to get a better picture versus 5 or 6 is what we do today. And that lets us find things that you might not otherwise pick up. Just to give you one example of this, if we could have the next slide, genome sequencing turned out to be powerful. 12 individuals learned something pretty important here. We had one person with a BRCA mutation. You'll see that about third one down. And then 3 people actually learned something that turned out was really pretty important. The RBM20 I mentioned before, that individual, fairly young, turns out their father died of a heart attack in the 60s. Aunt had one in her 50s. And we sequenced his genome right around the time his father passed away. And sure enough, he had a mutation in this gene, and then he did a follow-up stress echo. And sure enough, he actually has a heart defect. And so presumably this is the culprit. And he's on drugs now as a consequence. So this turned out to be an important finding. All the way at the top, 2X means there were two individuals that had a mutation in that gene, SDHB, which puts them at high risk for certain kinds of cancers. Or one of those individuals, fairly young as well, did a whole body MRI follow-up. And once you know it, they had early thyroid cancer that was removed and they were fine. Going further down the list, one person had 9 people are thought to be diabetic coming into the study. One individual turns out to have a mutation in the HNF alpha gene. And so they weren't a typical type 2 diabetic. They're actually a MODY mutation, which is very different. They tend to be treated differently from a typical diabetic. And so 3 people actually had phenotypes where actually had symptoms that were consistent with these genetic mutations. And so even though it was a minority of people that learned something from their genome sequence, 12 out of the first 70, if you're one of those 12, it's a pretty big deal. Now, we've actually gone on, I won't spend much time on this because I'm not sure if this is the right audience. Next slide. A lot of diseases, they're really not these simple BRCA kinds of diseases. They call them Mendelian diseases. They're actually a more complex disease. And these are thought to be due to variants, genetic variants of low effect. But if you get lots of the bad ones, you're at high risk. And if you get lots of the good ones, you're at low risk. And so the idea is that people these days do something called GWAS studies, genome-wide association, to try and find these. We actually think that way it works, but it's not very powerful. And it really only works for the top couple percent of genes. And I think that's because of 2 things. One is genetics isn't linear. And the other is that it ignores all the rare variants. And so we've come up, I won't get into details, with new machine learning ways of analyzing genomes that have been really quite powerful. And we've gone into 3 cases that we know are fairly genetic: abdominal aortic aneurysm, 70% heritability. There were 3 kind of questionable genes known, and we found 60 based on our new way of analyzing genes. For ALS, which is about 60% genetic, we took a GWAS study someone else had formed and went from 70 genes to 700 genes, really quite powerful, validate a lot of these, all these cases. And then lastly, severe COVID. Not when people get older, certainly their immune system gets weak, but even young people get severe COVID and can wind up in the hospital or die. And that thought to be genetic, and we did our new machine learning method. And if you're interested, you can look up some of these papers. We went from 47 known genes to over 1,000. So it's real. These new machine learning methods are very, very powerful. Okay, next slide. We think, actually, in the case of AA, we've gone on, I forgot to say, we built predictive models. We can actually make some level of prediction, come with a prediction score just based on your genome sequence. It is improved if you add some electronic health records. Looking over to the left, it's a decent score. It's actually potentially usable. And so we're rolling out some studies now. And we think this is powerful because for AA, it's only discovered when people's aorta bursts, or that's typically when it's discovered. And of course, they die when that happens. So having a good genetic score, we think would be very, very powerful. Next slide. So genetics is only one part of all this. We think other technologies are very powerful as well. And I won't get into this, but the imaging, the molecular, all wound up revealing situations. Even the wearables found things that weren't otherwise found. We'll talk about wearables a little more in a minute. So, one of the other things we learned by doing these deep profiles on people over time is that we could actually see how they're aging. So remember, these are just generally healthy people, but we actually see the molecular changes that are going on. And what we discovered is that everybody's aging differently. So the person 2 there in the middle, they're actually cardio-age, or their top pathways that are changing are the cardiovascular disease pathway. And the person on the left, their top pathways are pretty standard, actually. Metabolic pathway, the one shown here is the coagulation system. We think you need as few as 5 measurements within 2 years, and we can tell you how you're aging. And as I say, everybody ages differently. And we can group them into patterns based on the data we had. And we found 4 different patterns that are shown on the right. We call these ageotypes. And I know there are more than 4 patterns. It's just that this is all we're statistically empowered to find. Some people are kidney agers, other are liver, metabolic, and immune. So the rows are the different ageotypes. The columns are different people. And the people on the right, they're aging in all 4 categories. The box on the right, that person's aging in 3 of the 4, not much of a kidney ager, but aging more in liver, metabolic, and immune. And the ones on the left, the box all the way on the left, the one leftmost one's kidney and metabolic ager. And the second box from the left, that one's mostly kidney ager. So the point is we see how people are aging. And there are actually clinical markers associated with these ageotypes. So the information is actionable. And there are simple solutions you can try and do. We're actually trying to correlate these now that we have a bigger study to see what's going on. But we're for example, if you're perhaps an immune ager, you should take more immune suppressants like garlic, things like that. If you're a kidney ager, maybe drink more water. Liver ager, maybe don't tie one on so much, that sort of thing. So we think the information could be pretty useful. Next slide. One question I get asked a lot is how are you going to scale this? What we did is a research study, no question, collecting all that data over time. And we think it's quite powerful, but it's super, super expensive. And so people say, well, how are you going to scale that? And the answer is I'm in Silicon Valley, you form a company. So we formed a company called Cube Bio, and this is some of the first results we got. I forgot to say, but both in that case and this case, in the original study, the average age is 53.4, which is why I think we're finding things. Maybe find less if people are in their 20s. But saying it's an older group here around the same age. But nonetheless, even from the first less than 100 people, we found some pretty big things. Like starting from the bottom, we saw caught some with early ovarian cancer, prostate cancer. We even found early pancreatic cancer. And typically, no one line of evidence found this. That was true of our IPOP study as well. Sometimes it was imaging, sometimes it was genome sequence, but often it was the combination of things. Like you'll see some markers are off. The same time you see something doesn't look right forgot to say, these guys are also doing a whole body MRI. Now, in today's world, everyone will tell you, do not do a whole body MRI. And if we were in person, I'd ask you, why is that? And the answer is physicians, really 100% of them tell you, don't do a whole body MRI because you're going to find nodules. And it's true, men have them in the prostate, women in their ovaries, 100% of the time. And I say you should get a whole body MRI. It's not whether you have nodules, it's do you have any growing nodules? And the only way you know that is through the longitudinal profiling. So we think that's very, very important. Okay, so anyway, we again think that these big data measurements can be very, very powerful. And I'm pretty confident the prices can get a lot cheaper, especially with what I'm about to tell you next. So this is all fine. One of the things that's bothered me a lot is why do we go to a doctor's office for healthcare? Now, I get it. If you're doing a whole body MRI, nobody's going to have one at home, or unless you're super, super rich. But for many things, health monitoring can be done remotely. And so we've been working pretty hard on what I call Amazonizing healthcare. And there are 2 approaches that we're using. One is the wearables, and the other is microsampling. So starting with the wearables, there's thousands of these devices out there. Many of you, again, if we're here live, I'd ask you, I don't know if you're allowed to put your hand up in the chat. How many of you are wearing a smartwatch or a ring? Is there a way to do that? Maybe not. All right. Anyway, so if we're Zoom, you'd raise your hand or something. But the point out of all this is that these are getting quite popular. In the US, 20% of the population is now estimated 70 million people have a smartwatch. I myself am wearing 4 smartwatches. You can see my ring. I have an exposometer, a CGM that I often wear, and so on and so forth. And these are very, very powerful devices because they measure you 24/7. And so they can follow some pretty important parameters. Now, they all pretty much, for the smartwatches or rings, they all pretty much measure heart rate variability, and with reasonable accuracy, I would argue, at least resting heart rate. And then some of them measure skin temperature, some blood oxygen, some blood pressure, galvanic stress response, different things for different devices, and the accuracy can vary a bit with the devices. Even when the accuracy is off, we think they're so powerful because they'll be able to see shifts. So we started putting these on our cohort, and one of the things we discovered right away, next slide, is that we could pick up when people are getting infectious disease from a simple smartwatch and a pulse ox. And so this actually happens to be me. The backstory was I was helping my brother put up fences in rural Massachusetts. This was in 2015, and then I was flying to Norway through Frankfurt. And on that last flight, if you look at the bottom, my blood oxygen dropped, the blue dropped abnormally low, way much, much lower than it should for that jet. You can tell I measure myself all the time. And it never came back to normal when we landed. So the blue was less than the green. I also saw that my heart rate was up. It's low resolution plot at the top. And I later learned my skin temperature was elevated as well. So the point out of all this is that, and this is all pre-symptomatically, like I was seeing these things shift off. And then I got a fever off and on for the next few days. And I did then go to a doctor in Norway. I warned him it might be Lyme. He draws blood and sees my monocytes are up, my immune cells. And he says, all right, yep, you've got a bacterial infection. He wanted to give me penicillin. I said, no, I need doxycycline. I kind of warned him ahead it might be Lyme because of the timing, 2 weeks. And sure enough, he gives me he gave in and gave me doxycycline, and it cleared it right up. You do take it 2 weeks. And when I got back, I got tested, and sure enough, I'm Lyme positive. That's that positive sign at the end. And it just so happens I had given blood several days before I left. I measured myself a lot, and we looked at that sera, and it was negative. So I sera converted during that time. Very well controlled experiment. So the bottom line out of all this is that I first picked up my Lyme disease from a simple smartwatch and a pulse ox. So I won't share the data, but we went then analyzed all the data I had. I had 2 years of data. And it turns out I was ill 4 times during that period. One was the Lyme time, and there were 3 other times when I had viral infections, although one of them was asymptomatic. We know I was ill because my CRP reactive protein was up. And so every single time we could see that my heart rate jumped up in advance of symptoms. And we wrote an algorithm to do that. It worked on me, and it worked on 3 other people who were ill, and one of them got sick twice. Every single time we can see that they got ill because their resting heart rate jumped up. Well, so along has come COVID. Next slide. And it's a bit dated now. It's over a year old, but I don't know what the case numbers are in Europe. This is US cases. And these days they're still reporting about 40,000 cases a day. It's a vast underestimate because obviously people have largely stopped reporting. But, it's spreading around quite a bit. It's probably here to stay. And how do we measure when people are getting ill now? Next slide. Well, we use temperature, which is really a 300-year-old concept. Although these days you'll use an infrared one, will shine on your light. And if it's cold outside, it actually always gives a low measurement. I don't know about you, but mine tends to measure around 33, and they'll wave me in anyway, even after they measure this useless measurement, because you're not at 39. So the bottom line is we're using an old technology whose functioning is pretty questionable. Obviously, PCR and antigen tests are much, much better, but they're not the kind of thing you're going to do all the time. You don't run them, go up, wake up every morning and do a PCR test. It's just not practical. It's a little costly. So we think smartwatches have the potential to be quite powerful for disease detection. So next slide. So when the pandemic hit, basically what we did was we launched a study. We partnered with Fitbit, quickly got 5,300 people enrolled who first step was to see, can we see if anybody's getting ill from COVID with a simple smartwatch, all retrospective. And so we enrolled 5,300 people, 32 of which had COVID while wearing their Fitbit, and they had a diagnosis day and a symptom day. And this is our very first example shown here. The top graph is resting heart rate. It's a standard deviation plot. You can see they're marching along pretty normally. If you look closely, and this is where an arrow is useful, but look towards the end, see that green day there, day 48. That's actually when that person was diagnosed with COVID. And the red day before that is when they first had symptoms. Yeah, thanks. If you move your arrow over to the right of that. See the red bar there that's on the graph? Yeah, anyway, well, yeah, that's the mean summary. But anyway, look, if you look at the graph itself, sorry, the plot, yeah, the point is that this person's resting heart rate jumped up 9.5 days prior to symptom onset. Okay. And so, and presumably they were running around for 9.5 days, well, at least part of that time spreading the illness. Down below is a real-time detection algorithm I'll tell you about in a minute. Follows your baseline, looks for a jump up in signal. And so the right side shows a graph. It turns out it worked. This idea of this is all retrospective. We could tell when people are getting COVID in advance or at symptom formation, 80% of the time. So 26 of 32. And the median was 4 days prior to symptom onset. And it's reasonably good because COVID has a long pre-symptomatic period. Next slide. So we went on to build a system. We'd been building it even after my first Lyme case, and then obviously we accelerated when the pandemic hit. And so we have this real-time alerting system. It's a cloud-based system. It follows your baseline in a circadian fashion. And then if you jump above it for a certain duration or in certain magnitude, it'll actually send red alert. So we've set the alarms to go off roughly every 6 weeks. You can increase sensitivity, but then you get more alarms, or you can decrease sensitivity, but then you're slower at detecting when you're ill. You can sign up for the study. It's innovations.stanford.edu/wearables. Look at the bottom right. Or write to me and I'll tell you, but you can also put it in the chat, innovations.stanford.edu/wearables. And it turns out it works. Basically, next slide. So what you do is you click on your app every morning and you see whether you're at baseline or whether you jump up. And if you jump up, you get a red alert. And this is one of our first cases. You can see when this person first had symptoms, they were actually getting red alerts for 3 days prior to symptom onset. They were diagnosed positive the next day after symptoms. So, and that is the median time for the real-time alerting. It's about 3 days prior to symptom onset. And same number, it works about 80% of the time. And that number is held true. I know we can get that number better. We have a hard time getting a stable baseline on certain people. Next slide. So it works on Apple, it works on Fitbit. It should work on all devices because it's an anomaly detection system. And it even works in asymptomatic cases. So here's a Fitbit case. We have some for Apple as well. It's probably hard to see, but there's a purple date there, sort of at the bottom of all those red lines. Yeah, that's it. So basically, this person was diagnosed that day, but they were getting red alerts for 2 weeks prior to no symptom, prior to diagnosis. And so, again, pretty clear signal. And again, presumably they were spreading it around without knowing it. And so we think this could be quite powerful. Next slide. I should point out, though, it's not specific for COVID, not surprisingly. Other infections will trigger this as well. On the right, you can see even mycoplasma infection can do it, other influenza. This is just looking at COVID negative people. Turns out the number one trigger of red alerts is workplace stress. Your heart rate does jump up when you have a workplace stress. Other things trigger it too, like intense exercise, really drinking a lot, excessive drinking. Not 2 drinks for dinner, that won't do it. But if you really tie one on, your heart rate will be up for a few days. Travel, we know, triggers it. So a lot of this you can contextualize. It's pretty easy to tell if you got a red alert what it's due to. But if you're just sitting around and not, you don't think anything should be happening, you're getting red alerts, something's up. You're either psychologically or physically stressed. Next slide. So we can also then, here's my personal example. And I'm one of several cases. It turns out it's more sensitive than an antigen test. So here's my personal case. I can show you another one that looks just like it. I happen to have a family stress event. These are actually real dates. I don't mind telling my story, but there's a family stress event. I had to travel, those 2 red alerts. Mostly you get green days. And then I was getting ready for a trip to New York City. You can see where it says 10. That's April 10th last year. I wake up and I'm just a touch congested. And I'm wondering, is it due to allergies? What's going on? And so I do an antigen test, top left. And where it says T, you can see I'm negative. I didn't, no positive tests at all. I look at my smartwatch and I'm a bright positive. So my smartwatch detection. or smartphone, red alert. So what I do, I got on the plane anyway, and that was a big, big mistake. And I fly to New York City, and the next day, in order to go to the meeting, you actually have to get a COVID test. And once you know it, I'm bright positive. So I spent a whole week in New York City in a tiny little hotel room, all because I listened to my antigen test and not my smartwatch. So we think your smartwatch is more sensitive than an antigen test. You only need 2 beats per minute to see this jump up in signal. And so again, it's not as specific. We don't know. If I didn't know it was COVID, or I wouldn't have got on the plane. But the point is that, yeah, it is quite sensitive. I know with more data, we'll be able to tell the difference between COVID and workplace stress. Their signals should just be quite different. That's why I want you to sign up for a study. Next slide. So we also can pick up long COVID signals from this. People with long COVID tend to elevate a heart rate post-COVID. Next slide. We think this is just the tip of the iceberg. We've done some machine learning to see if we can get features from a smartwatch that'll set up predictive models for clinical tests. And so on the left side, you'll see hematocrit, red blood cell count, hemoglobin. We actually can get a pretty strong signal from smartwatch features for these things. It's not a clinical grade measurement, but it's good enough to see if a heart rate's jumping up. And remember, you're wearing these things continuously. And likewise, you'll see in the middle where it says A1C and glucose. That's fasting glucose. You can actually get a signal around your fasting glucose from a smartwatch. It has to do with dryness of skin and other things. So the bottom line out of all this is that, again, there's a lot of information in these devices that can be very, very powerful for your health, following your health. Okay, next slide. So the other area that we're pushing very, very hard on is continuous glucose monitoring. And these have become quite popular lately. We got started on this one. They're mostly being used for type 1 and type 2, insulin-dependent type 2 diabetics. We started putting them on normal people. People are thought to be healthy and pre-diabetics. And what we discovered, if you look in the middle, is that some people do have pretty good glucose control. Some people are actually moderate spikers, and some people are severe spikers, the ones at the bottom. And so we wrote algorithms to classify people into glucotypes of low, medium, and high spikers / fasting glucose, if you will. So then the other thing we discovered almost right away is that different people spike to different foods. Next slide. This is some work from Aaron Siegel who found the same thing. It turns out some people spike to bananas, other potatoes, some to pasta, some to white bread, some to brown bread. We're all different. And in parts due to the microbiome. That was shown by Aaron. And if you look at the graph in the middle on the top person, that person spikes to a banana, but doesn't spike to a cookie. Probably the only person on the planet doesn't spike to a cookie. The one at the bottom spikes to a cookie, but not a banana. And so again, people are spiking to different foods. Next slide shows some of our data. We found some people spike to bread and peanut butter, others to protein bars, which show carbs. 80% of people spike to cornflakes and milk. That stuff's nasty. If you don't remember anything from my talk, don't eat cornflakes and milk. I don't think they should put warning labels on that stuff. Probably worse than smoking. Okay, next slide. So we think these remote monitoring devices are going to be very, very powerful. So can we extend this into biochemistry? Well, we've been working hard. We actually spent about 6 years, and I noticed one of the sponsors is Neoteric. So we're very happy they are. A great relationship there. And so what we played around with a lot of different technologies. Like I say, spent about 6 years on trying to find something where we can get pretty stable analytes measured, things that would stay stable. We can measure fairly deep numbers of them. And so we settled on two things. One is Neoterics and the other is Tasso. So they're a sponsor as well. And so basically the idea is you do these little pricks. You take tiny droplets of blood, you FedEx it to our lab, and then we do our deep omics profiling, lipidomics, metabolomics, proteomics, and then targeted assays. And in the end, we can measure about 2,200 different analytes, a lot of which are pretty high value. And so it works pretty well. Next slide. We spent a lot of time measuring stability. This sounds like a simple experiment, but we took, we had samples for 5 different durations up to, say, 120 hours. I forgot how many days that is, quite a few days, number of different temperatures. You wind up, it's a lot of experiments on 2 different people, their collections. And so what we discover is most proteins are actually pretty stable. Only a few percent go off, even at the most extreme. And then for metabolites, they're also pretty stable. Some do go off. And lipids, maybe a little bit less so. It depends on the lipid. But we know which ones go off and which ones don't. We can even put corrections on that. Next slide. So then we did some really fun experiments. We took 32 people and we had them all drink this shake, Ensure Shake. I don't know. You guys are mostly UK, I imagine. But this, I don't know if I don't personally like it myself, but you can, yeah, we measure people before and then 30 minutes, 60 minutes, 120, 240 minutes after drinking the shake to see what the response would be. And what we discovered, this is not a surprise, hundreds of molecules change. They were fasted initially, then they drink this thing. Hundreds of molecules shift. You can see the bottom left. And a lot of key molecules shift. What's nice is that we can quantify this and measure it. So we can see C-peptide, insulin, a lot of pretty important peptides, even that incretin called GIP-1. We can measure that from this microdroplet sampling assay. Same, we can measure some of these inflammatory markers. Leptin went down. That actually surprised me. In hindsight, I think I know why. So the cool result we found is the next slide. What we discovered is that everybody reacts to this thing differently, the shake differently. So each box is a different person. And this is just showing carbs, but I could show you the same for amino acids, for free fatty acids, what have you. Now this part will get a little bit trickier, but look where the arrow is. That's about a third of the way over. If you look at the box there, you'll notice that that person, it's probably hard to see the name, but you'll see their carbs plummet. If you see that brown box going down, all three carbs for this person, they tend to go as a group, all plummeted after drinking a shake. You go over 3 later, their carbs plummet, that person. So just scan your eye across. You'll see some people who don't have much of an effect for carbs. Other people, their carbs go way up. Other people, their carbs go way down. The one on the left, all the way on the left, that person drops their carbs after drinking the shake. So they're all drinking the same thing, but having very, very different responses. And then if you look at the very bottom part, this could get a little tricky too. Look at the right side. We group these into 5 types of people. Gray is the average. You see those little gray balls. There's a brown ball that's kind of, well, if you go on the left, group 5, I don't know if you can put your arrow there, Phil, but bottom line is that brown dot, that means yep, you got it. Their inflammatory markers drop after drinking the shake. They're less than average. So the shake is anti-inflammatory for them. But you go to the person next one over, group 4, and look at the same brown ball, that goes higher than average, meaning it's pro-inflammatory on those folks. And so the exact same shake for some people is suppressing inflammation, for others it's pro-inflammatory. And again, everybody's having a very different response, which is kind of cool. So next slide, we put it all together. Now, this is where I see this going. This is a pretty extreme form. This is an individual who basically gave these samples every hour for 7 straight days, all the waking hours, not once at night, but generally not at night. And they did the microsampling again for a little over 7 days, 98 samples. And at the same time, they were wearing a CGM, a continuous glucose monitor, and a smartwatch, and doing food logging. So you can see thousands of molecules change at the bottom, the multiomics data, those patterns of the molecules that change. And you can correlate it with what they're doing, their activity, from their wearables and that sort of thing. And I guess the movie's not working here, but it's kind of unfortunate. The right side, what you would see is that we do time lag correlations. We see which events are associated with what other events in a correlative fashion. That's probably the simplest way to describe it. And this is not a surprise. Next slide. We'll see thousands of changes, biochemical changes that associate with each other, as well as with heart rate, steps, and glucose. And, to give you a simple example, if you walk, your heart rate goes up, and we can measure that. It's about a 1-minute time lag. But we can measure that for all these things, again, get very quantitative. Now, if you look at the middle part where those plots are, what you're supposed to see is there's a pattern between CGM, meaning glucose, and C-peptide, this insulin peptide, and they're highly correlated. That's known. But what's nice is you can measure in this person. Turns out to be a 10-minute lag. You can see exactly, you can quantify it. If you go to the bottom plot, we discovered some new inflammatory markers that go up that might actually be involved in mediating the immune response. The FLT, can't quite read the bottom part, but that gene's known to promote an immune response, and we can see it comes up about, I think it's about, excuse me, 15 minutes after the glucose. What's kind of cool is the top one. Alpha-synuclein is it's involved in dementia, Parkinson's, what have you. We can measure that with this microsampling assay. It's pretty cool. And it does correlate with glucose, and it has a fairly regular pattern. I think it's actually correlating with other stress events as well. And so we think that's going to be really important to measure. Imagine you're at risk for dementia or Parkinson's. You probably want to follow that and see what you can do to push that off. Much like you want to avoid diabetes by avoiding your glucose spikes, I'm pretty sure that you want to avoid getting alpha-synuclein spike if you're at risk for dementia or Parkinson's. So that's how we're seeing to use this technology. And again, we can get some very, very deep dives on this. So where's all this going? Next slide. We can pull on all this data, the wearable data, the omics data, your microbiome, what have you. We pull it all in onto your smartphone with something called My PhD app. You can display this at whatever resolution you want. Next slide. And then what we think is that we'd like to be able to use this kind of information to better manage people's health. And so this is where I see this going. This is Mike Snyder's world. I see a world where people are getting their genome sequenced before they're born. And then together with biochemical and wearable measurements on the top right, you'll be able to better manage people's health. You actually build personalized AI programs around people to see what shifts them and what keeps them, fairly stable health-wise. And so again, one question, how do we scale this? Well, it's the same theme I told you earlier. Next slide. We do spin off. Oh, sorry. This is a team that did this. Something got out of order here. I had its peak at a really large wearable team that were working furiously to try and, detect COVID and other illness. Still have a big team on it. And the microsampling team shown below. And the next slide just shows virtually everything I've done. You can spin off into, well, we don't spin everything off. A lot of it is, everything I showed you was research. But we can try and apply it and get it out to the community by, we think, scaling. I think academics, they're great at discovery. They're great at proof of principle, not very good at scaling. It's all these companies in the box. I found quite a few companies, but the ones in boxes actually directly spun off the work I described to you. And so we can use, for example, we have a long COVID company that can, better diagnose, we think, and treat long COVID based on some of the things. We have a microsampling company spun off and so on and so forth. So the January Eyes, the CGM company that we spun off as well. So again, we try to actually bring these out to the community. And the ultimate goal is a transfer of healthcare so that you can take a lot more responsibility for your own health rather than wait for your doctors. So that's the story I wanted to tell you. Happy to take questions if you have any. So again, thanks very much for having me here and listening to my story.
Thank you, Mike. It was a really interesting story. Let's just have a quick look at questions. We've got a question here. It would be interesting if we could compare this information with profiles from centenarians from the Blue Zone project. So more of a statement really than a question. Anything that you're aware of there, Michael?
Well, we are very interested in that ourselves, so I couldn't agree more. What do their profiles look like? People have sequenced genomes of the so-called wellness, elderly, and the most striking thing is the absence of some like Alzheimer's related mutations, that sort of things, absence of the high-risk cardiovascular disease things. So I do think there's a wealth of information in the elderly, and I couldn't agree more. I would love to see, in general, if you open up a person who dies when they're super old, people live to be 100, their organs are all aging kind of equally. So they don't seem to have a blind spot like many of us who get a heart attack or cancer, and that's what bumps us off. Okay,
thank you. We've got a question that says, has this been recorded and will it be available? The answer is yes. It is being recorded. and it will be available on the website where you registered, and there'll be also be a link to it from the patient-centric sampling interest group website. So yes, that will be available. Someone asking, please share the link to the software that reads the iWatch data and performs the analysis. So I don't know, Mike, whether you're able to share that link.
Yeah, I'm going to put it in the chat. I hope you can see it. Let's see, innovations.stanford. Please do sign up. We are trying to perfect our, I know we'll be able to tell, like I say, much better than we can currently. We weren't even working with heart rate variability, which we know drops. I just got a shingles vaccination, super cool. We could see my resting heart rate went up, my heart rate variability dropped. So it really does work. And I didn't show you the COVID vaccination. We have similar data. So hopefully you can see that in the chat.
That's appeared.
Great.
So
love to have you all sign up. So please do.
And I guess we can share that after the event as well, potentially.
That'd be great if you wanted to send it out to the audience.
Yeah.
Group.
I have a question, and it's something that the patient-centric sampling interest group is kind of all about, and that is, we see lots of great technologies, great approaches. The problem is, how do we break through to get these actually adopted? And I just wondered whether you have your own personal thoughts on that. You know, how do we turn what you're showing us into something that's readily available, recognized, and potentially reimbursed?
Yeah, no, great. I'd say there's two issues. I have a lot of things around that. One is, at least especially in the US, it's the whole financial incentives are not aligned, meaning nobody pays to keep you healthy. They only get paid when you're ill. So that's why we practice sick care, not healthcare. So I think we need financial incentives to keep people healthy. Like maybe you should get a discount if you wear a smartwatch and walk your 10,000 steps a day. And you can tell when people are cheating and they put it on their dog and stuff because the patterns are pretty different. So I think you can set up incentives like that. I think, and also, or maybe get family groups to do it. So I think we need to get this better. I think getting employers involved. I mean, even at Stanford now, they have wellness programs. A lot of big employers are starting this. So I think that's one way to get it going. The other is, you know, what do you do about physicians? And the problem with physicians is they're very negative on this. When we started sequencing healthy people's genome, every one of them said, that's a bad idea. All you're going to do is make people, turn people into hypochondriacs. They're going to be really mad at, you know, they're going to do millions of dollars of follow-up tests, and nothing could be further from the truth. Actually, now, in fact, most physicians are accepting it. But I can tell you pretty much all physicians don't want you doing whole body MRIs and don't want you they don't want wearables on people, because people show up and say, hey, what's going on with my watch? And I think that they just haven't learned to embrace it. But in the future, having watches tell you whether, you know, what your baseline is, and when you shift from that baseline, it's going to be standard stuff. So they really should they should be embracing it. They just don't have time to embrace it. The other part I'd like to say is that, you know, nobody has time. This group knows it better than anyone else. No one knows it has more time to take care of you than you. You have a lot more time than the physician. You know, in the US, an average physician sees a patient for about 15 minutes. And that's it. And they're trying to make a whole interpretation around you in 15 minutes. I think this is where, you know, ChatGPT types of things will be much better than a doctor. They won't replace doctors. They're meant to augment them. Obviously, there's a lot of garbage in there right now. But I think we can pull in information in a much more useful fashion the way we do it. And so we've got to educate doctors. We got to set up systems. You know, what I showed you is really clunky. We pull in data lots of different ways, but that'll be automated in the future. And we now have AI programs built around me. They all run in the background. So a long-winded answer, I'll just add one more comment on that, which is I view health much like your car, where, you know, your car, you have sensors on your car. You have a dashboard that's always reading off your health. You don't stare at your dashboard all the time. You do when you care about speeding or running out of gas. But that's how I see health in the future. You'll have sensors on you, like watches, ultimately implantables, that will follow your health in the background and tell you, you know, hey, something's up when things go off.
Yeah, thanks. So along those lines, we had a comment. Someone loved the idea, but their experience with 23andMe is that physicians did not know what to do with the information. So my question is, do you think there will be a hurdle for clinical acceptance of this information, given med schools may not be teaching this level of diagnostics? And I think you kind of answered that already.
Yeah. So I think that's right. I think we have to educate doctors. We have to get them to embrace new technologies instead of being fearful. Most physicians say, they come from the do no harm. Well, doing nothing is harmful. But I also think that 23andMe's this polygenic risk score stuff, which is pretty hard. That really only works for the top couple percent of people. Stuff we do, like with the metabolite profiling, you can see when things are off. I already, told you a few examples like infectious disease, things like that. So I think the kinds of profiling we do is a lot more accessible, which is why we found so many health discoveries. We did find a lot from the genome, don't get me wrong, that's important. But I think it's only part of the whole picture you're trying to get when you're trying to predict health risk or tracking people's health. So it's a great question.
So we have another question around clinical laboratories not having moved to microsampling due to their instrumentation requiring larger volumes. So this person's asking, do you have any recommendations to engage the labs and their instrument manufacturers? And that's a big question we all have.
It's a great question, is right. Yeah. So I think what we have to do is show clinical utility. You run these things out as research studies. I view it as analogous to genome sequencing. There was a lot of pushback when genome sequencing came out. And then people started showing its utility and figured out how to better use those. Still pretty expensive. The nice thing about the microsampling stuff is it's not that expensive. And so I think it'll be able to move into the mainstream quicker. It does need robust protocols, built around it, which I know a lot of groups are working on, including some of the sponsors here. And so I think those that will help accelerate it. I do think it formats a little bit better into the way medicine's practiced than genomics did. So I think its adoption could be faster. And I think the idea of doing these longitudinal profiles, people will get and realize how important that is. So I, maybe I'm being pretty Pollyannish. The other nice thing is that, when kids are born, people do metabolite tests, usually about 50 markers in the US. Not sure how many is in the UK. And that's done with mass spec, the same technology. Now, we use pretty sophisticated ones for us, the $1 million instruments, whereas the ones that use in clinical lab, they're probably about $300,000 instruments, maybe, depends. The point out of all this is that it's somewhat of the same format. It's more expensive, but I think we can get it adopted. I hope quickly, because I think this is the future. And I love the idea of this, monitoring at home. And the time when people are ill, they don't feel like going to a doctor. And that's exactly when you want to be testing them.
Yeah, absolutely. I think it's an exciting time. And as a community, we're trying to figure out how we break down the barriers to enable this kind of thing to at least be an option for people. And that's kind of why we formed the patient-centric sampling interest group, because we realize that moving that next step, whether it be for healthcare, whether it be for wellness, clinical trials, drug testing in sport, it's kind of difficult changing the status quo.
It is, yeah.
And how we move that forwards.
In the US, the wellness is the way to go because it's less regulated, at least for the moment.
Yeah.
You can run these wellness programs, give information back. And it's a lot easier to run. And that's useful because then it helps establish what is useful, what's not. Yeah, setting it up as a clinical test requires, as you know, it's pretty highly regulated. So you've got to follow all the right procedures. And that makes sense too. Yeah.
So thank you for sharing your email there with someone, Michael. You were multitasking well there, I saw.
Yeah, I guess I've learned to do this in the Zoom era.
So
great questions, everyone. So,
a last question, maybe. There are many gaps in the knowledge of capillary blood. Many analyzers do not have approval for the clearance of capillary blood samples.
That's a great question. Yeah, let's see.
What analyzers did you use for your trials involving capillary blood?
Yeah, well, we're using we're using Luminex assays for the targeted ones I told you about, for the incretins and some of the C-peptide, I think. But most of the omics profiling is done with a mass spec. It's a thermo instrument. You may be familiar with the Orbitraps that are quite, sophisticated. They're very high sensitivity and they have high resolution. So, and they are research instruments. And you're raising a good point, because this is whole blood we're pricking, whereas the medical establishment's pretty used to plasma. And we did do a lot of analysis there. I didn't show that, but there's about 80% strong correlation of analytes in plasma and blood. Some things are different, and that's not surprising, because obviously the cells in your blood have a lot of metabolites and things like that. But a lot of it does correlate, and that stuff you can use then straight up. But I think what one will have to set up a whole reference system built around whole blood that's different from what we're currently doing with plasma. Will it lose sensitivity in certain areas? Almost certainly. But will it gain sensitivity by being able to measure hundreds, if not thousands, of analytes? Absolutely. I think the whole thing's a win in the end. And our company, YOLO, if any of you are in the States, is actually, they're measuring several hundred analytes just with the exact same tests I'm showing you. And that one's with a Tasso-type device. And it's just been very powerful for getting a good molecular portrait of a person's wellness. Great question.
Yeah, I think we're just going to wrap up now, Michael, if that's okay.
Sure. Great questions.
It's been a great session. What I'd just like to make a plug at the end here is for your host today, the evidence base and the patient-centric sampling interest group. So the patient-centric sampling interest group was brought together by passionate individuals from around the world who are interested in how we can work across organizational boundaries to make the use of these alternative blood sampling and analysis technologies a reality for healthcare, for clinical trials, for wellness, drug testing and sport, and anywhere else where they may be able to be used. And we've got a number of people working on a number of projects at the moment, giving up their time. If you're interested in being involved in the group, please, on LinkedIn, just search for PCSIG. You'll get a company, please follow, and a group. Please ask to join. There's currently just under 250 people in our group. Someone's just asking what your company is, Dr. Snyder. Oh, you've already answered that. You're on it. So for those in the pharmaceutical industry, you may be interested in joining the AAPS group. There's some contact details there. These slides will be made available on the evidence base and on the patient-centric sampling interest group. So don't worry if you didn't get these links. And I'd just like to thank all our sponsors again, without whom we couldn't do this. Our next event is in September, on the 27th of September. We'll watch out for adverts about it on the evidence base, on LinkedIn, and on our group. And we'll be running we're planning to run about 10 of these webinars a year. So if you have ideas for webinars, please contact me or anyone else involved in the patient-centric sampling interest group. With that, thank you very much, Michael. This is a really fascinating webinar. Looking forward to more to come, and just hearing how you're going to break down the barriers and change things in the way we do health, Michael. It's really, really fascinating.
We're working on it.
Thank you for everyone for taking part today.
Bye all.
Bye.